So we've talked about eSpOT-ON. Our collaboration with Gene Therapeutics has been one of the most productive partnership in our CRISPR portfolio. And what emerged is hfCas12Max. It's a purpose-built engineered version of Cas12I. Has exceptional fidelity, a broad PAM flexibility, and a compact size ideal for delivery. And so Gene's team rigorously benchmarked it alongside Cas12A and other Cas12I variants. And hfCas12Max consistently outperformed editing across editing efficiency and specificity. And so just like eSpOT-ON, we're not just offering another enzyme, we're offering a vetted, scalable solution co-developed with Gene that they're already in the clinic with and have been producing lots of great data. And I'll just briefly highlight some of that data here. There has been several talks, and Gene's been going around to multiple different conferences, sharing a lot of great data. And so I'll just highlight a little bit of that here. And so we've had hfCas12Max for around a year now. It's a little bit smaller, about 20% smaller than sbCas9, which makes it ideal for delivery via AAV and other size-limited systems. It also recognizes a 5' TTN or TN PAM, shown on this heat map here in the green, benchmarked against other Cas12 variants, providing broad genomic access. And so the enzyme was evolved through a directed evolution strategy that targeted key DNA interaction domains, resulting in an editor that's both highly efficient and highly specific. And so it's been validated in multiple different formats, RNP formats, mRNA, and AAV, and it's shown excellent performance across T cells, liver, and CNS applications. So let's look a little bit at the data from hfCas12 in real therapeutic models. And so in primary T cells, they achieved 90% knockout of the tract locus and 80% viability post-editing. So that's not just efficient, it's well tolerated, which is critical for cell therapy workflows. And importantly, it demonstrates effective multiplexing and including simultaneous knockdowns of TRAC and B2M at the same time, consistently with high editing rates. And so once again, we also know about we want to know about how it performs in vivo. So we'll just take a look at some of that data here. And so using LNPs to deliver hfCas12Max mRNA and guides, researchers achieved a significant reduction in cccDNA and HBsAg2 key markers of viral persistence. And this suggests real promise for curative interventions for chronic hepatitis B, where reducing the amount reservoir of infected cells has been historically difficult to do. And so importantly, this wasn't just effective, it was also safe. So mice tolerated the treatment well, and no signs of liver toxicity or weight loss across the study, even after high dosing. And so this model underscores what we've seen in other systems. hfCas12Max performs in vivo with clinical grade precision. And while not shown here, data from ALS models further reinforce this. Editing ATXN2 in TDP43 transgenic mice reduced neuroinflammation and toxic protein aggregation with meaningful functional improvements. And so combined with earlier T cell data, where we saw high TRAC knockout, strong viability, even with multiplex edits, hfCas12Max continues to prove itself as a flexible and high fidelity platform across therapeutic success. And so building on this, Gene's been very active in their therapeutic pipeline, advancing hfCas12 into multiple different clinical developments in their HG302 program, utilizing hfCas12Max and DMD, and has entered the muscle clinical trial. So early data from the first dose patient indicates very favorable safety and tolerability with no serious adverse events reported. So moreover, initial assessments really show improvements in motor function and suggesting potentially therapeutic benefit.